Hello everyone, this is chapter 2.2 and under discussion in today's videos are functional groups and IR spectroscopy. So let's get started. So there are going to be two videos in this lecture. The first will cover a diverse array of functional groups which are going to be essential to your understanding of organic chemistry. The second video will talk about the physical basis of IR spectroscopy and how biologists and chemists alike use this as a diagnostic tool. We've already met the hydrocarbons in our earlier lecture, chapter 2.1. Hydrocarbons are organic compounds containing only carbon and hydrogen. They include alkanes, alkenes, alkynes, and the aromatics. The latter three are considered different types of functional groups as they contain a reactive centre. But what is a functional group? Well, in organic chemistry, functional groups are specific substituents or moieties, just a fancy way of saying a group of atoms that are bonded together within a molecule that may be responsible for the characteristic chemical reactions of those molecules. So the same functional group will undergo the same or similar chemical reactions regardless of the size of the molecule that it is part of. Now, alkanes are not considered as functional groups. Instead, an alkane is a compound that lacks functional groups. The functional group in an alkene is the carbon-carbon double bond, that of an alkyne is the carbon-carbon triple bond, and aromatics are cyclic structures. For this course, it's sufficient to know that benzene is a planar molecule and that it is fully conjugated, has alternating pi bonds, which are delocalized. Though you should recognize this last group as a functional group because of its ability to undergo particular chemical reactions. Before we continue, I wanted to tell you about one further sub-classification of certain types of functional groups. So when you have an sp3 hybridized carbon that is connected to a functional group that we're calling X, now this is typically for alcohols and for the organic halides, then you have to think about what the carbon that is attached to that X group is also attached to. So for example, if the first group, which is in red, note how the carbon is only attached to one other carbon. Therefore this would make the X a primary group, whereas the blue functional group that's represented by the letter X, the carbon that X is attached to two other carbons. And finally, when we have our tertiary group, so this is the X that's marked in green, then the carbon that X is attached to three other groups that are connected to carbon atoms. And this is a way of representing primary, secondary, and tertiary groups. Sometimes these are denoted as 1 degree, 2 degree, or 3 degree, and they mean primary, secondary, and tertiary respectively. Here's our first functional group, the alcohols. On this slide there are some natural products. See if you can identify the class of the alcohol that's indicated, that's primary, secondary, or tertiary for each molecule. Pause the video and resume when you are ready. Okay, so citronella's alcohol is primary because the carbon bearing the alcohol group is attached to one other carbon. Cholesterol's alcohol is secondary because the carbon bearing the alcohol group is attached to two carbons. And yes, finally, tea tree oil's alcohol is tertiary because that carbon bearing the alcohol group is attached to three other carbons. So the other type of alcohol that gets its own slide is the phenol. And phenols are alcohol groups that are directly attached to a benzene or an aromatic ring. The old name is carbolic acid, which also speaks to another difference between this type of alcohol and those that we encountered on the previous slide. This alcohol is quite acidic, and we'll talk about that in future lessons. Seeing as I've included some natural products already, here's another one that contains this alcohol group, and this is capsaicin, and it's the chemical that's found in chili peppers, and it's what makes them hot. So it's colourless, odourless, and oil-like, but if it comes into contact with your skin or mucous membranes, you know all about it. let's survey some more of the functional groups that we'll encounter in Orgo 1. When oxygen forms two bonds and they're both to carbon atoms, then you get an ether. These are often used as solvents. The polarity and the hydrophobic portions, especially THF and 18 crown 6, make them superb solvents in which organic synthesis is performed. And this is because they can dissolve an array of non-polar and polar solutes. 18 crown 6 even dissolves metal salts. Now, another special type of cyclic ether that you'll meet later in this course is called an epoxide, and this contains a three-membered ring where one of those atoms is an oxygen. These are very strained and therefore are highly reactive, unlike the other ethers. We'll talk about these in later chapters. Organic halides, i.e. organic molecules containing fluorine, chlorine, bromine or iodine atoms, are very simple functional groups. These have a rich chemistry and feature heavily in this course as they are a common substrate, often the subject of transformation into other functional groups. This small selection of organic halides we see here achieved notoriety as they were designed with good intentions in mind. Hexabromocyclodecane was originally intended for use as a flame retardant. CFCs, such as the refrigerant Freon, were intended to keep your fridges cool and your houses cool in the AC system. And finally, dieldrin is an insecticide used to kill insects that grow on food. All of these substances are now banned by the Environmental Protection Agency because of their harmful effects on the environment, health, or both. Yet these compounds do provide an opportunity to survey your understanding of our earlier classifications and to add a couple more. Please identify any primary, secondary or tertiary halogen groups on these compounds. Pause the video and resume when you are ready. Okay, HBCD contains only secondary bromine atoms. Now, Freon can't be classified as either primary, secondary or tertiary because of the lack of any other carbon atoms. When two halogen atoms are connected to the same carbon, they are described as being geminal. Hence, the fluorine and chlorine atoms are both geminal. Finally, the chlorine atom attached to the alkene on dieldrin is not primary because of its connection to one other carbon atom. When halogen atoms are connected directly to an alkene, they are said to be in the vinylic position. There is one more geminal dichloride in dieldrin, seen here, and the other two chlorine atoms represent a tertiary functional group. There are 5 kinds of amine. We'll start with the simplest, ammonia. Now, every other amine is based on this framework by having replaced a hydrogen atom for a carbon. And this is what allows us to classify these functional groups. When we replace one of the hydrogen atoms with, say, a methyl group, we call this a ethylamine. And this nitrogen then is attached to one other carbon and so becomes a primary amine. Next, we replace another hydrogen with a second carbon atom, another methyl group, and it becomes dimethylamine, which is a secondary amine. We do the same thing again, and we get trimethylamine, which is a tertiary amine. Finally, because of the ability of the lone pair of the nitrogen to form a dative covalent bond, i.e. it shares both of its electrons, nitrogen ends up with a formal charge and can form a salt. Now, these are often referred to as ammonium salts, or you may see the phrase quaternary nitrogens. Amines have a very characteristic smell. The smell of fish actually is the amine triethylamine, and there are 2 other much more sinister smells that are associated with death and decay. And I think the name putrescine kind of says it all. So if you can conjure up the worst possible smell, then this is the smell of decay. Whereas actually you add 1 carbon unit and you get the smell of a dead body. So this is called cadaverine. So these are probably the worst kind of amines. These are the ones that smell of death, However, there are some good amines, and we'll talk about those on the next slide. But for now, what two types of amine are present in these three molecules? Pause the video and resume when you're ready. Okay, so triethylamine, well, that's a tertiary amine as the nitrogen is attached to three other carbons, but both putrescine and cadaverine contain primary amines as the nitrogen of each amine is only attached to one other carbon. Well, that's enough about death, so what about the living? So here are two functional groups for the price of one. These are amino acids. So we have an amine group and a carboxylic acid group, which we'll be discussing later. But for now, these are both essential amino acids in that we get them from our diet. So both essential and non-essential amino acids are involved in protein synthesis, and without these, your body struggles to grow and repair. So for example, there is a metabolism disorder called phenylketonuria, or PKU, that results in a decreased metabolism of phenylalanine. And untreated, PKU can lead to intellectual disability, seizures, behavioral problems, and mental disorders. Whereas valine, if you have a deficiency in this amino acid, this is also marked by neurological defects in the brain. So these are incredibly important amino acids that we get from our diet. Next, let's talk about the carbonyl functional groups. The carbon-oxygen double bond between those two atoms is referred to as a carbonyl bond and is itself not a functional group. What the carbonyl carbon is connected to, this is what determines the functional group. So the first functional group of this type occurs at the terminus of an organic molecule when the carbonyl carbon is connected to hydrogen. Let's think about some of the functional groups that are present in these molecules. Pause the video and resume when you think you've identified them. The simplest aldehyde, methanol or formaldehyde, is a preservative of biological specimens, and this is the only aldehyde that has hydrogens on both sides of the carbonyl carbon. There are no other functional groups present. The carbons that are connected to an aldehyde can be aliphatic, that is, contained branched or straight chain alkanes like ethanol, and ethanol is a major industrial precursor to many important molecules. But the branched or straight chain alkane is not really considered a functional group. Or they can be connected to aromatic groups. In this case, the benzene group is a functional group, and benzaldehyde is a representative aromatic aldehyde used in flavorings and fragrances. The last example shows glucose, which is an aldose sugar. This contains alcohol functional groups, and typically you're more used to seeing glucose as its cyclic version. Nonetheless, written in this way, glucose, like all the other examples, contains the aldehyde functional group. But what if we replaced both atoms adjacent to the carbonyl carbon with carbon? This time round, you get a ketone, and these are internal groups, never found at the end of a molecule. There are three different ketones on display here. See if you can identify any other functional groups. What do you notice about the first and the third ketone? Pause the video, and when you are ready, please resume. Well, propanone, the middle ketone, is another useful solvent, but it also contains only one functional group. That is the ketone itself. However, both R-carvone and S-carvone contains two types of alkene. That is an isolated alkene at the bottom of each molecule and an alkene that is conjugated, which I've highlighted here. In order for the alkenes to be conjugated, it just means that there is another pi bond within one carbon's distance. Lastly, the first and third molecule are the same, but these are stereoisomers. They are non-superimposable mirror images, and if you remember, we call these enantiomers, but we'll be discussing more of these enantiomers in Chapter 5. Let's move on to the last set of carbonyl containing functional groups. The last functional group that contains the carbonyl bond are carboxylic acids and their derivatives. So to be a carboxylic acid, the carbonyl carbon is attached, with the exception of methanoic acid, to one other carbon atom and a OH group at the end of the molecule. And these are typically weak electrolytes that are able to donate the hydrogen ions into solution, hence they're acidic. There are some other functional groups in each of these acids. See if you can now identify them on the basis of our discussions concerning the alkenes on the previous slide. In the last example of abscisic acid, I have also given part condensed structural formula for the carboxylic acid. Pause the video and resume when you have your answers. Okay, so ethanoic acid doesn't contain any other functional group except the carboxylic acid group. The second is a byproduct of anaerobic respiration. So when you get that stitch in your side or a pain in your side after running for a prolonged period of time, it's usually because of the buildup of lactic acid in your muscles. And this molecule also contains an additional alcohol functional group. And if you indicated that this is a secondary alcohol, then well done. Lastly, abscisic acid is a plant hormone, and this is what causes the leaves to fall from trees. This contains a conjugated ketone, as highlighted here, and there are also 3 bonds in conjugation to the acid, as highlighted here. Now, just as it was possible to replace the groups that are attached to the carbonyl carbon with oxygen, then we need to think about replacing the hydrogen with either alkyl groups, or we replace the oxygen itself with nitrogen, and we get carboxylic acid derivatives. I'll discuss these in turn in a moment. When the acidic proton, that's the hydrogen ion of our carboxylic acid group, is replaced by a metal or other positive ion, I'm thinking of ammonium here, we get a carboxylate ion. Often, carboxylic acids that contain large hydrophobic chains are converted into these carboxylate salts, especially when they're needed to be in water. Any idea why that may be? Pause the video and have a think. Resume when you are ready. So if you were thinking about increasing the solubility by taking advantage of the ion-dipole interactions with water, then that's absolutely spot on. The second derivative of a carboxylic acid is what happens when you replace the acidic proton with a carbon side chain. And these can either be aliphatic or aromatic. And you get a molecule that's known as an ester. And the ester is a functional group as indicated here. These are often used for flavourings and fragrances. Lastly, we have this molecule called aspartame, and this is an artificial sweetener that is 200 times sweeter than regular sugar. And this contains the secondary amide, as the nitrogen is connected to 2 other carbons. However, there are 4 other functional groups present in this molecule. What are they? Pause the video and resume when you think you have an answer. Okay, so we have the carboxylic acid at the terminus of the molecule, and this is, it's disguised in terms of being a condensed structural formula. We also have another primary amine, which is highlighted here. We have the benzene functional group, which is highlighted here. And although the man is in the way, that is actually an ester group. So OMe, if you remember, Me is a CH3 group. One of the most commonly made mistakes by students is they will look at a carboxylic acid and they will say that, oh, this contains an aldehyde and an alcohol together. But here we have to remember that atoms attached to the same carbon count only as a single functional group. So if there is a carbonyl and an OH, this in itself is a carboxylic acid. So to follow on from that, the amide, as you see here indicated on the right, is not made from an aldehyde and an amine because both of those bonds, that's the carbonyl and the amine, are made to the same carbon, and so therefore this counts only as a single functional group, that is an amide. Final nitrogen containing functional group is called a nitrile, and this is any organic compound with a carbon triply bonded to a nitrogen. The picture of the gloves are here because the gloves are made from a polymer which contain this functional group.
Concludes part A.
In this video we're going to learn about the physical basis for IR spectroscopy and how we can use this to probe molecules as part of their characterization. So, firstly, a molecule will be in its ground state, and when we shine IR radiation at it, some of this energy will be absorbed and some of it will be transmitted. IR, like other forms of electromagnetic radiation, carries energy, and specific energies of IR are absorbed by molecules. So once this energy has been absorbed by a molecule, we can see that it's moved to a higher energy level, and it's possible to measure what has been transmitted and what has been absorbed. However, unlike in modern chem, where we focused on electrons being absorbed and emitted in atomic absorption and emission processes, IR only causes changes to bonds in what were called vibrations or rotations. So we're going to have a look and see what that means in terms of the bonds that are involved. Here's a diagrammatic representation of a molecule, which we can represent as two atoms connected by a stick, which is representing our bond. However, we need to adapt this picture slightly, as the stick model really represents the bond as being this static thing, whereas actually if we connect our two atoms and think of them as two masses that are joined by a spring, then we get some different behaviour. And what does that mean once the molecule has absorbed our IR radiation? Well, it means that the bonds stretch and they contract, like they would if they were connected at the two ends of a spring. And the energy that causes the bonds to vibrate came from the absorption of the IR. And that's got to be at a specific frequency and with a specific energy. Remember that energy and frequency are related in a directly proportional manner. Just for completeness, imagine the bonds behave like springs. Can you think of two factors that might affect the bond vibrations? That's the stretches and compressions of this bond model. Pause the video and have a think. Hopefully, this diagram concerning Hooke's law might provide some clues. The force needed to cause a spring to stretch and contract depends on the strength of the spring, which is an analogy for the strength of the bond. If we think about the length of the spring, a short bond is going to require more energy to keep those springs extending and contracting at the same rate. We know that the hybridization of the atoms, and hence the bond that connects those atoms, has a direct effect on the bond length. So therefore we can say that pi bonds, so triple bonds for argument's sake, will require more energy to keep them vibrating than the corresponding double and single bonds. Of course, we have to compare like for like. So in this case, our two atoms would have to be of a similar size. But speaking of which, the other factor that affects the vibration of the spring will be the two masses that are attached to either end. And so a lighter atom is going to vibrate much faster if it is connected to a heavier atom. When I was taught this concept, I was told to imagine a spring connecting a ping pong ball to a much heavier ball. Think of a bowling ball. To pull the small ping pong ball, let it go. And of course that ping pong ball, our hydrogen atom, which is really what this is representing, is going to vibrate many times. And the bowling ball, well, doesn't move. So of course the input of energy, though it's me, is going to continue at the same rate if I continually pull back that ping pong ball and let it go. So this is representative of the specific amount of energy that would be supplied by IR. So speaking of which, this is something that I've said a few times throughout the course of these slides. Bonds vibrate with certain frequencies, and of course it's bonds that make up different functional groups. So if we can cause those bonds to vibrate, we can use IR as a diagnostic tool to probe organic molecules with and see what type of bonds they contain, and hence understand what type of functional groups they contain also. IR is part of the electromagnetic spectrum, and so what's useful about this is that the long wavelengths or low frequency, low energy waves can excite certain bonds within organic molecules, whereas conversely, shorter wavelengths of IR with higher frequency and hence higher energy will excite other certain bonds within organic molecules, and it's that we can take advantage of. So as a quick cartoon model here, when we hit this molecule with a continuous range of IR radiation, then depending on the chemical makeup of this molecule's bonds, certain bonds will absorb certain wavelengths of energy. Thus, by observing the wavelengths that are missing, i.e. transmitted like those with frequencies nu1 and nu2, we gain insight to what bonds are in the molecule. This concludes video 2 and chapter 2.2.